This way of making squalane via industrial biotechnology can help save or avoid the killing of millions of sharks each year to support the squalane market. And this way is so efficient that we can produce the world's annual supply of squalane simply by harvesting sugarcane from an area of 1 kilometer squared. That's less than 1% of the land area of San Francisco. 1 kilometer squared of sugarcane is enough to produce the world's supply of squalane via industrial fermentation with our optimized, tailored microorganisms. In addition to squalane, there are a large number of other products that we have commercialized and continue to manufacture. 12 of them are shown here on this slide. And the way that we develop the technologies for manufacturing is, as I said, our proprietary lab-to-market platform, which starts with automated DNA design and automated strain design, and moves on to automated strain engineering. The strains that are built are then tested initially through high-throughput screening, and the very best strains then are sent for testing and fermentation. Throughout all of the testing that we do, the screening and testing and cultivation, of course, we rely very heavily on a variety of different chemical analytics. And in all of the work we do, we capture all of the data and the metadata to enable and facilitate and accelerate learning by both machine learning and human intelligence. We take that learning then and we go back to the design phase and we redesign strains and go around and around the circle until our strains are good enough to begin intensive process development and manufacturing. I'm going to talk about the DNA and strain design and also the strain engineering, and then I'll hand it off to Mike, who will talk about the rest of this wheel. So my focus and specialty is strain improvement. And what does that mean to improve the strains? What are we asking our strains to do? The objectives are, first, high fermentation yield and productivity. In essence, we want our cells to be able to make lots of product fast from minimal inputs. We also want to enable high recovery yield and low purification costs, which means that our strains should avoid making byproducts and impurities. And finally, we want to have long fermentation runs to enable high fermenter uptime, make the best possible use of our capital investments. And in order to enable long fermentation runs, it's important for the cells to be able to tolerate any toxic product or intermediate. They must remain healthy and vigorous in the fermenter for long periods of time. And in some cases, it's important for us to engineer the cells to resist evolution, because evolution in some cases tends to lead to lower producing but faster growing cells, shown here in the blue line, where performance falls over time, which is bad. And of course, we want to enable the cells to be able to maintain high levels of performance in the industrial fermentation for a long period of time. So how do we do this? How do we improve the strains to try to achieve these objectives? Much of our work is devoted to rational, what we call rational approaches, where we think deeply, we use modeling, we read the literature, we rely on expertise to take approaches like metabolic engineering, enzyme discovery and enzyme engineering, and transporter engineering to control the influx and efflux of metabolites in and out of cells. But biology is really complicated, and there's a lot about biology that we just simply don't understand in sufficient detail to be able to model and predict. And therefore, we found it's critically important to complement the rational approaches with random approaches, including random whole cell mutagenesis, adaptive laboratory evolution, and genome scale diversity libraries. I've mentioned before this classical engineering cycle, or the iterative design, build, test, learn cycle, where we go around and around this circle until we have strains that are good enough for manufacturing. And indeed, we've found in most cases, we have to go through many rounds of this cycle. And in the end, our manufacturing strains are as different from the starting microbial strain as a Chihuahua is from a Great Dane. And this is true both in terms of the behavior or phenotype of the strain, but also in terms of the genetics and genotype of the strain. So this is a major undertaking. Our manufacturing strains are very highly evolved, very highly tailored in order to maximize the conversion of sugar into the targeted chemical products. So we have invested a great deal of time and effort and money in improving the design, build, test, learn cycles, in building a R&D pipeline that makes these cycles faster, smarter, higher throughput, and facilitating deeper learning. So this cycle starts with automated design. We find the optimal route, the optimal biochemical route from sugar to our target molecule. And then we translate it into DNA instructions. We've built computer programs and computational systems to enable and facilitate this. The transformation of sugar into the target molecule involves a series of biochemical reactions called a route. And we sometimes represent, when we're depicting these routes, we represent the molecules as circles and the reactions as lines connecting the different circles together. This is a map, a metabolic map of metabolism from Kyoto University. You can see that it's very complex. And what we've done at Amyris is built a computational atlas of metabolism that comprises not only all of the metabolites and biochemical reactions that are known in our microbial host, for example, in yeast, but also all of the metabolites and biochemical reactions known from all organisms throughout the world. And this then enables us to create, in essence, something like a Google Maps of metabolism, where we can say, we want to start from glucose and we want to end at trans-beta farnesene. Press the button, hey, machine, tell me the fastest way to get from A to B. It takes only moments to identify an optimal biochemical route, and the computational program not only tells us what is the fastest route, but it also gives us certain specifications or information, metrics about the route. For example, in this case, it's comprised of 19 reactions, and the maximum theoretical yield is 23.8%. After we've elucidated the desired route, we use a programming language for DNA that we've devised called Genotype Specification Language, or GSL, which can translate a higher order of abstraction, a higher order, an abstract specification of a genotype, like that shown here in this colorful line on the left, in the middle of the left panel. It can translate this high order specification of a genotype into a detailed 7-part, several thousand base pair sequence like that shown on the right. Through developing this automated design platform, we have moved from a condition in 2008 at Amyris where it would take one individual 3 weeks to create 20 designs, to a situation in 2020 where, with instant route finding and DNA design, the computer can design 1,000 different genotypes in 10 minutes. Once we have the designs, then our automated strain engineering group can translate those designs, can actually instantiate the designs and make thousands of strains each week. First, we build the DNA parts. DNA parts are amplified through polymerase chain reaction, or PCR, or in some cases, we synthesize the individual DNA parts by chemical synthesis. We have over 200,000 DNA parts stored in our freezers, so we can often retrieve those parts from the freezer and use them that way. And then we assemble those DNA parts. We make 1,500 DNA assemblies per cycle, and we can initiate a cycle every 2 weeks. Once we have those DNA assemblies, we integrate the DNA into the chromosome or chromosomes of the host cells using designer nucleases to trigger high-efficiency multi-locus integration. The transformation components often include multiple donor DNAs and one or more plasmids that encode a nuclease. The nucleases then cut the chromosome in one or more locations. For example, here I'm showing 3 chromosomal locations that are simultaneously engineered. The nucleases cut the chromosomes, thereby triggering homologous recombination or repair of the cut site with donor DNAs whose ends match the ends of the chromosomal cut sites. And in fact, we often use 2 or more donor DNAs per chromosomal locus, which allows integration of very large multi-gene DNA constructs. Here's a video where I'm showing the echo chamber, which is an integrated robotic platform that we've built, a custom-built robotic platform at Amyris. And I'm going to stop talking for a moment and let you watch the video and read the words on the screen. So we have a number of these custom integrated work cells, both for strain building and for strain testing, which Mike will talk about in just a moment. And these types of custom integrated work cells using acoustic liquid transfer and miniaturization, for example, in 384 well plates, has enabled us to increase throughput by 1,000-fold and decrease cost per strain made by 100-fold over the past 12 years or so. So now I'm going to stop sharing my screen and hand the baton off to Mike, and Mike will talk about the rest of that wheel that I showed earlier.
Great. Can you see my screen?
Yes.
Yes. Thanks, Kirsten, for a great introduction to our pipeline, and thanks to the organizers for inviting both myself and Kirsten to speak today. So I'm going to start today by talking a bit about test, and specifically what is test, what do we do in test, what are the challenges that we faced in testing our strains. I'll talk a bit about our capabilities in fermentation, analytical chemistry, and bioanalytical chemistry, and then dive in a bit deeper to the science of high throughput screening. Slides won't advance. 1 second. I apologize, my slides won't advance. Can you see my screen again? Here we go.
Yes.
Okay, good. So the overall goal of test is to identify winners from the strains that Kirsten's team creates. And at Amyris today, we produce 7 million strains per year across multiple product lines. So we have about 15 product lines going through our R&D department, and we have to test out of those 7 million for all of those different products that we're producing. So then test job is to take those 7 million strains and sort through them and say, in high throughput screening, which ones do we want to pass on to our most limited resources in fermentation, and then either put them back through the R&D cycle for further optimization, so going through the design, build, test, learn cycle again, or pass them into manufacturing so we can produce products. So this is, of course, this is one of the challenges is the number of products that we support and also the number of strains that are coming through per year. But it's a bit even more complicated than that, in that we have an immense amount of product diversity. So Amyris's mission is to be able to produce sustainable products. And that means that we have to be able to have our test platform to support many hundreds of molecules. So as soon as the business team decides that we'd like to go after a given target, we are ready to go. So I'll first talk in detail about our fermentation and analytical suites. And then, as I said, I'll go on to onto the high throughput screening platform. So our fermentation suite is cutting edge. We have 156 bioreactors, roughly evenly split between glass vessels and ambers. And so those are at the 250 mil scale up to the 2-liter scale. The whole pipeline is supported with a custom LIMS system. So we've built this so that we can scan the feedstock and scan the samples coming out of the bioreactors. And all of this is automatically ported into our LIMS system so we can evaluate the strains. Of course, because we have such a high number of bioreactors being run, and these are continuously run throughout the year, we also are supported by a very advanced automated sample preparation set of robots that allow us to take the fermentation broth, process it with high accuracy and precision, and also confidence, because they're barcoded, that the samples that we're measuring have come from the fermentations from a specific time point and then can be analyzed, and that data can be fed back through our LIMS system to then analyze those strains. Supporting the fermentation platform is our bioanalytical suite and our analytical chemistry suite. So the traditional analytical chemistry tools that we use are traditional liquid chromatographs and gas chromatographs, looking at either feedstock concentration, products that are produced, or byproducts that are produced. And of course, this is very important because we need to know that we're producing our product in high purity so that we have high confidence when we go to manufacturing that we can make that product, purify it, and it can be used in the application that we've designed it for. Also supporting strain analysis is our bioanalytical chemistry suite. And so that's looking and being able to interrogate the different strains and the genotypes of them to understand the phenotypes. So looking at the transcripts, the proteins, the metabolites, to look in detail at the engineering and say, did the engineering have the desired effect? And if not, what do we have to do to go back and re-engineer that strain so that it performs as expected? The technology is continuing to advance, especially on the bioanalytical chemistry side. And so we're supported by high-end tandem mass spectrometers, which is state-of-the-art today. So all of these investments and our track record and experience in producing multiple products, to date we've produced 13 products at manufacturing scale, allows us to have the results that you see here, where we're able to very confidently promote strains from our lab scale bioreactors. So those measurements are on the x-axis here, and they match very well to the fermentation performance at manufacturing scale. So on the left-hand side, you see these numbers for farnesene, which was our flagship product that we used in biodiesel and we're using today for the production of squalane, where you see yield on the top and how well the 2-liter correlates to the 100,000 liter scale. And similarly, on the bottom on the left side is the productivity and how well that matches between the two scales. On the right-hand side, you'll also see that we have 8 other molecules and how well that yield and productivity matches for those 8 other products. And again, this is not only through the investment in the platform, but also through our vast experience now that we've built up over the years. So I'd like to take just a little bit more time to dive in more into the science of high throughput screening because it's quite complex. In high throughput screening, only 1/2000th of the strains that are generated to the sources that Kirsten spoke of are promoted to fermentation. And the technology in this field is moving very rapidly. So I'd like to just dive in a bit deeper there. So finding winners in high throughput screening is extremely challenging. I like to compare it to finding a needle in a haystack, where the needle and the haystack are changing over time. So we often test our strains in 96 well plates. And what we're trying to do is mimic the conditions in a 96 well plate to what we would find in a commercial scale or a bench scale bioreactor. And of course, the genotype is changing over time. And so that means the phenotype of the strains are changing over time. So we're always operating with we're operating one step behind in our culture models that we have because of that. So just to kind of outline the differences between these two scales, so screening at a 96 well plate scale versus the manufacturing scale, we're trying to identify strains with improved performance at 1/100 millionth the volume, with no real-time feedback or process control as you'd have in a bioreactor. Of course, we'd ideally like to measure yield, productivity, biomass, and sugar consumption, which are the traditional metrics that we use to evaluate our strains at manufacturing. In reality, we're generally measuring titer as a surrogate for yield and productivity. But these are endpoint measurements where we've gone through sugar exhaustion and then we're reading the titer at the end of the culture. Biomass is pretty straightforward to read in 96 well plates, and sugar consumption, because they're endpoint measurements, we're assuming sugar is at 0. We do all of these tests with the requirement of high throughput, so on the order of less than 1 second per sample and low error. And of course, we're supporting all the molecules in R&D, and we're doing this while the genotype is changing, and therefore the phenotype and physiology of the strains are changing. We've recently published on the challenge of high throughput screening and the best knowledge that we have to date on how to do this at Amyris, and you can see the reference at the bottom of the slide here. But just to illustrate the complexity, in the left graph, you can see if we were optimizing a strain with anywhere between 2 and 14 genes being optimized, and we had 4 different promoter levels driving those genes, the library size increases combinatorially as we go across that. And so we said, if the standard is the standard that Amyris has, which we have 156 tanks or the ability to analyze 7 million strains per year, and we wanted to do an experiment in 1 week, how many genes would these different, or how many genes could we optimize at once using either only tanks or plates, or in the case of microfluidics, so droplet microfluidics. So you can see here that very quickly we reached the limit of even what we can do in 1 week in plates with only an 8-gene pathway. And so this really drives the argument that we need throughput to be able to engineer biology, because the diversity is vast. And as Kirsten said, we only know so much about it. And so we have to rely on taking many chances and trying many times and sometimes failing to be able to learn. The same argument can be made with protein engineering, where you can see the number that in plates, the number of targeted residues for just 1 mutant across those residues versus double and triple mutants very quickly goes beyond what we can even do at Amyris today. So we know and we've learned that faster assays accelerates the strain development progress. And this is an example from our sweetener program, where we had started off, and you can see the progress graph here, where we started off using a very low throughput assay to be able to measure our strains. So it was a traditional chromatography assay, roughly 15 minutes per sample. And we realized very quickly that we were limited because we couldn't test the library sizes that we needed to optimize that strain, or optimize a sufficient number of genes at once to then be able to go back and quickly learn and redesign. So after introduction of the high throughput mass spectrometry platform, you can see that our sample library sizes went from somewhere in the neighborhood of 10,000 samples per, well, in this case, it's per quarter, to in the 50 to almost 100,000 samples per quarter. And the yellow line here represents the yield at manufacturing scale, which was the major metric we were trying to optimize for at the time. And the slope of that line indicates progress. You can see as we started to learn, we saw rapid advancement in strain progress. Now, of course, this is only one part of the challenge in high throughput screening. As I mentioned, we're often culturing in 96 well plates, and what we're trying to do is find the best way to optimize that culture to best represent what we can do in these highly controlled bioreactors. And so there's many different conditions that we can look to try to mimic in many ways to do that, whether that's limiting oxygen availability or maximizing oxygen availability, controlling temperature, feeding strategies, so that would be delivering sugar in a controlled fashion in these 96 well cultures, or even onto product removal or even retention. We've worked with volatile compounds at Amyris before, and that's often a challenge because they'll just evaporate through the seal of the 96 well plates. You can look in detail here, but suffice to say there is a lot of work that goes into optimizing these 96 well plate cultures, and a lot of work to continue the ability to pick the winners and confidently see them perform at scale in fermentation. So at Amyris, we're always trying to do better, and Kirsten's team has continued to build out the way that their capability to advance diversity, improve diversity, and give us even larger and larger libraries to screen. And so in high throughput screening, there are many next generation readouts that we have and that we've been investigating. So when I think of different analytical assays and what we could use in high throughput screening, I classify them in terms of, or I think of them in this way. I look at them in terms of throughput, which here is on the y-axis, and then measurement quality or information density, which is on the x-axis. And oftentimes what you find is that the higher the throughput you have, the less quality you get or the less information density you get, and vice versa. So at Amyris to date, we have all of the technologies listed here in yellow that we use for strain screening. And are investigating the different technologies that are cutting edge, shown in blue here. And I'll talk about each of the blue ones and where we think they are in terms of state of the art. So the mantra that we use is the future is plateless. And today it's almost plateless, meaning that we are still screening today in the lab in 96 well plates, but we see that as a major limitation because it takes up such a footprint in terms of just culture space, but then also you have extensive robotics to be able to manipulate those 96 well MICRO-TITER plates. But how do we go faster and how do we remove the burden of plates? So this first technology is a bridge. It will still be using 96 well plates, but it allows us to go tenfold faster than what we can do today. Kirsten mentioned the acoustic liquid handler that they have in biology. This has been interfaced with a mass spectrometer, so you can transfer droplets from a 96 well plate into a mass spectrometer and then get an analysis. What this allows us, though, is, again, it's tenfold faster than rapid fire, which was about 10 seconds per sample. And it allows us to go to between 1 and 3 seconds per sample. All of this with the information density and measurement quality that you'd find with traditional mass spectrometry. So we've done an evaluation on this piece of equipment, and we actually published an application note with SciEx, the instrument company, and we anticipate bringing this in-house this year. So we're really excited about that. But again, we're still limited in this case by culturing and by extracting and processing in 96 well plates. And so these other two technologies, one is called the beacon and the other is traditional droplet microfluidics. We've investigated each of these to see how our strains perform in these. So the beacon is an instrument that's produced by Berkeley Lights. They're also in the San Francisco Bay Area. And what this instrument allows us to do is to culture our yeast on a chip. So this chip here is about the size of the United States quarter. So think of this as 2.5 centimeters in this distance here. And this chip contains what they call pens. So each of these are individual wells where we can grow a strain of a single genotype. And the technology exists to be able to transport only one cell in each well. And so we did an investigation with Berkeley Lights on their beacon, and with a molecule that is naturally fluorescent. And you can see here, the green is actually the fluorescence produced by that molecule, the fluorescent signal, and you can measure biomass at the same time. So what this allows us to do is to measure productivity, which we traditionally haven't had the ability to do at small scale. When we look at the bioreactor, the correlation between bioreactor and beacon, and the beacon itself, you can see that we have very good correlation between our strains at the small scale to the bioreactors at bench scale. So we're very excited about this technology, again, because it's giving us a real-time productivity readout, and it removes the need for all the laboratory space. The key limitation of this technology today is that we are stuck with molecules that are naturally fluorescent, or we need to engineer a biosensor or some other way to transduce product titer into a signal that's readable. We published on this. It recently came out in a Lab on a Chip journal, and so you're welcome to go see the full detail there. The third technology that I think it's even further away than the beacon, but it is very exciting to us because it takes us into the millions of strains per day being able to be evaluated. So that's just an immense throughput, much more than what we can do today. And so this is where cells are encapsulated and grown in oil droplets, or aqueous, there's aqueous, of course, media on the inside, and the oil droplets encapsulate the cells and they grow there. And then they're sorted by lasers. They're read by lasers and sorted into high and low producers. We're still investigating this. I don't have any data or paper to present yet, but this is something that we're definitely keeping our eye on, and is something that we hope to institute in the next few years at Amyris. So what I hope that we've shared with you is that we are delivering on the promise of synthetic biology. Kirsten and I have both been at Amyris for many years, and I know we're both immensely proud and continue to want to work on this technology development, because it's what drives us, right? It's what brought us to Amyris in the first place, which is using our technology to do good in the world. We've shown you a bit about our highly automated systems that we have. It's both in robotics and computation. We've talked in detail about high throughput technologies that we have and those that we hope to have in the future, and also how we use very large data and designed experiments to continue to maximize the amount of learnings per cycle. Using all of this, we've launched 13 new products over the past 10 years, and we continue to want to show to continue our track record of success in technology transfer to manufacturing. So we can go beyond those 13 products that we have today and to the many, many, tens of, 50 and on to 100 and beyond. So with that, I'd like to thank first of all, all of you, the audience, for being with us today, and of course, to acknowledge so many people that we have at Amyris that have been working on technology development and making our pipeline run. And we'll take any questions or comments. Thanks.
Okay, thank you, Michael, and thank you, Kirsten, for very interesting presentation, very clear. Congrats for that impressive work. And we have now time for questions. Let me see here in the chat box if we have already questions. I have one. It's from Gordon, and I will read the question. Is not directed for any one of you. You can decide who is going to answer. And the question is quite large. Let me see. The market seems to overlook Amyris strains engineering capabilities in Facor or Flashier companies like Zimmergen and Ginkgo. Sorry, I don't know if I'm spelling good. Can you take a moment to delineate Amyris from the other two firms?
Yeah.
I can answer.
You want to go
this.
first, Kirsten? Yeah.
Sure. So Zymergen and Ginkgo are younger companies taking the same sort of technological approaches that Amyris is taking to engineering biology and using engineered organisms to make products. And I think the biggest distinction between Amyris and the other two firms is that Amyris has, from its inception, had a much greater emphasis on process development, process engineering, scale up, and manufacturing. And you can see that in our track record of success. Over the past 10 years, we've launched 13 new products. Zymergen does not yet have any commercially available products, and Ginkgo partners with a number of companies, but they have not focused on or emphasized developing process development, process engineering, and manufacturing capabilities. They've not built a manufacturing facility. And so that's a big distinction between them. What we've found between Zymergen and Ginkgo on the one hand and Amyris on the other, and what we've found at Amyris is that it's really critical to have under one roof people who specialize in developing the strains and people who specialize in developing the products and people who specialize in manufacturing, having those specialties under the same roof, but interacting in a highly collaborative and collegial way. And that's been critical to our success, and that differentiates us from Zymergen and Ginkgo.
Yeah, and I think there was a bit on the market and why they value one or the other. You know, it's hard to really understand the motivation behind the market, but I would say that the flashy side that you brought up is something that, you know, it excites investors when new companies come out. And so they're looking at the dream of the company rather than at the actual fundamentals of it. And for Amyris, in Amyris's case, you know, we went through a hard time that we've come out of, and now we have many brands and many products, and we're continuing to show our revenue and even profit growth. And so, you know, it's only a matter of time before the market recognizes that. But, you know, that is a frustration, especially when we were going through our hard time many years ago, that the market wasn't recognizing the true capability of the platform.
Okay, I'm going to the second question. It's not too clear, but I can try to reformulate the question. The question is, are bioplastics a possibility with this technology? I think is asking about the platelet technology that Michael was talking about. No, the person who's...
I think maybe it's asking, can we use this technology to make bioplastics? And indeed, this technology has been used to make bioplastics. It's not something that Amyris is currently focused on, but the polylactic acid that's used in the compostable plastic cups and those sorts of products, those are made using this type of technology. So lactic acid fermentation is used to make lactic acid that's then converted to polylactic acid to make those compostable cups. Also, there are some other companies, for example, I think Genomatica is focused on one of the projects that they're currently undertaking is a bio-based replacement for nylon. That's not something that Amyris has been focused on, but this technology is applicable for that type of product.
Yeah, yeah, and the difficulty in those molecules can come from the actual price that you can sell them for in the market. So I think, you know, the way that we think about difficulty of our products is first of all about the cost that they can fetch in the market, and then also what the requirements are on our microbial factory, cell microbial factory, to be able to scale that. So the way that we thought about that for farnesene is that how far are we from the maximum theoretical yield of that cell factory, and do we have to get? So if we have to get 95% of the way there, then, you know, we really need to think about that, because it'll be a very large investment. If the cost is a bit higher and the requirements on the yeast and on the downstream manufacturing process is less, then we'll often go after those things. And we go through that rigorous evaluation for all of our products before we undertake R&D on them.
Okay. Other questions are coming. I have here another one from Gary. How much faster can your throughput be than it's currently? I mean, at this point, are you expecting to increase your high throughput capacity? Gary is asking, is 10 times possible to increase this capacity in high throughput you have currently?
Sure. So today, I'm just doing some quick math. We have the ability to screen roughly 20,000 strains per day on average. And that's in the current platforms that we've been talking about, the rapid fire mass spec. So certainly with just the next generation mass spec technology, the analytical measurement can certainly go 10 times faster. But then the ultimate ceiling of that technology is the droplet microfluidics, which is millions of strains per day. Now we haven't, we've worked with external collaborators on this and we've seen their ability to get to that throughput. But in our hands, we don't know exactly, but we know it's at least 1 million is the floor, and it could be as high as 10 million or more when we have a mature technology in hand. But again, as I said, measurement, the quickness of measurement or the throughput of the measurement is only part of the equation. The other difficulty with droplet microfluidics is going to be making sure that they are representative or they give good enough data so you have confidence in what you see in the readout that you see. But certainly we can go tenfold for sure. And then beyond that, I guess that's going to be another at least 100 to 1,000 fold more in the next 5 years, I'd say.
Mm-hmm. Okay, great. That's great. Other questions are coming. For instance, Gordon is interacting a lot with many questions. One of them is also my curiosity. What are you thinking about next years or 5 years or 10 years? How much you are excited, or how many more molecules are you expecting? You were saying 13 products in 10 years. And what about the next 5 or 10? What are the expectations? And if you can talk also about applications, what have you guys in mind about that? Thank you. Gordon is asking this, and me also.
Yeah.
We are accelerating our ability to launch commercial products and new manufacturing technologies. So I believe that John Mello, our CEO, has said that we are undertaking efforts to expand our pipeline and our throughput so that we will be launching in the coming years 4 to 8 new products per year. That's what we're working toward.
Okay. That's a good goal, indeed. Yes, more questions for this time is Gordon again. Do you think that Amyris could be hypothetically become a B2B platform or foundry as gene composition? It is being discussed as a future pursuit. B2B platform.
Oh, for strain engineering, it sounds like.
No.
Well, we do have B2B relationships. We have historically had a number of partnerships with the leading flavor and fragrance companies in the world, for example, FERMENTATION, Givaudan. Those are B2B relationships. But we want to take great care not to be essentially a CRO or a contract research organization, but make sure that we maintain a substantial fraction of the value share of the work that we do, and that we carefully vet the opportunities that we go after. There are so many opportunities for using this technology to make new bio-based products, and we want to make sure that we carefully evaluate, as Mike was saying before, what is the cost-benefit equation? What is the cost of doing the R&D development work, and then the benefit? What's the market size? What's the cost target? What's the total addressable market? What's the revenue and margin we can expect to get, and how quickly? And so we are being very careful and rigorous in evaluating what new products and project opportunities we go after.
Yeah, and I think I'd add to that in that we are also, you know, as a business that's still becoming profitable, we are also being very careful to think about what we do on our own, meaning the products that we launch on our own, because then the value to us is much higher. And then we carefully consider also on the flip side, which products we want to work with someone externally for. So sometimes that mental or that equation or that thought process is generally around, do we have experience in launching products or producing products of that particular sort? And if so, then generally we might try to do that on our own. But if it's a new market or a new type of molecule that we're going to make, we'll oftentimes partner with the company because they have a better understanding of how to get the product into the hands of the consumers better than we would. But that's the way that we think about it in addition to the technology. Yeah.
Okay. More questions here. We still have some minutes, 2 minutes or something for completing the session. So you still have time to make questions or chat here in the box, or you can raise your hand. From Andrew, we have here also an interesting question. What was the most challenging product or molecule that Amyris has ever scaled to date? What was the more challenging one? And I can complement the question about what is the most limiting step? Is it the strain design, or is it the screening of the winner? Overall, what was the most challenge step and product that you ever had so far?
Sure. And Kirsten, I want you to talk about farnesene because you led that program for so many years. On the limiting step, biofine for sure.
Biofine.
In screening, I mean, we are always limited in our fermentation capacity or bench scale fermentation capacity. And we have a project today, an internal, what we call an infrastructure project, so not a molecule project, one where we're trying to make our plate to tank prediction better, meaning going from the titer plates that I talked about to the fermenters to always keep that, above 90% confidence that the strain is going to do better, because bench scale bioreactors are the best read that we have on how a strain will produce. However, they're also really costly. And even with 156 bioreactors in-house today, we're still limited by what our projects want by probably 800. They'd like 800 more fermentations run per month than we can today. So there's a lot of work that we're putting into that, augmenting that capacity, and I believe that's always going to be the case. But yeah, Kirsten, I'll let you talk about biofine because that's definitely your project.
Yeah, well, I think they're different classes of challenge. In some ways, farnesene was the most challenging product, but there are other projects that we've gone after that were more challenging in other ways. So farnesene, we went after farnesene initially for use as a biofuel. Yeah. That would be a whole other seminar, how we went after it as a biofuel and why we're not selling it as a biofuel. But we were fortunate in choosing our biofuel target, the fermentation product, farnesene, very wisely, because farnesene is useful as a feedstock for chemical conversion to make a wide variety of other products. So today we're manufacturing large amounts of farnesene. It's going into vitamin E, it's going into car tires, it's going into squalane, it's going into a number of destinations. So it's a great chemical feedstock. And as we were pursuing that project and thinking that it would be a biofuel, the major challenge was driving down the cost of production to well under $2 or even $1 per liter. And in order to achieve that requires reaching yield and productivity in the fermentation that is beyond what is even theoretically possible, the upper theoretical limit for the pathway that is naturally found in the microbes. And so what we needed to do in that case is essentially invent a synthetic central carbon metabolism that was far more efficient in enabling the cells to convert sugar and oxygen into farnesene. So we surpassed the maximum theoretical yield of farnesene by implementing a totally different central carbon metabolism that was far more efficient in the microbes. So that was extremely challenging, and driving down the cost of production of farnesene was extremely challenging. That's one type of challenge we've tackled. Another type is, in the case of the sweetener, that is a very complex, extremely long pathway with a lot of enzymes whose selectivity or specificity needed to be precisely engineered. So that's an entirely different type of challenge that we've tackled.
And I think adding to that, Kirsten, on farnesene, by the way, there is a paper that's been published in Nature. I don't have the reference with me, but you can look up Kirsten's last name for sure, and that would be there to look at that. It was a great amount of work that the company did. For the sweetener, the challenge, I mean, Kirsten mentioned many of the primary challenges there. There is also, we had to engineer a transporter to be able to transport the product itself selectively rather than the precursors that were related chemically. And that was actually critical to the project because otherwise the cells just stalled and basically died because they filled with the sweetener internally, but it wasn't being pumped outside of the cell. So that was something that was a big challenge. And that was one of the first molecules that we did beyond terpenes. So Amyris, when we started, we're engineering strains to produce terpenes instead of now, in this case, it's a glycosylated terpene. Mm-hmm. Thanks, Dominic. It looks like Dominic just shared the Nature paper. Thanks, Dominic.
Yes, we still have a lot of questions, but I have to ask to the host if we should close the session or should we continue. We have a lot of questions, but we have to accomplish the time of this session. I would like to ask to the host if we should continue, if we have some more minutes, or should we continue?
If you wish, please do.
Okay, let's select one or more, two, and to close. Let me see. People are curious about your current targets, but I don't know if you can reveal too much to us. I was seeing here one question about that, about your current molecules on pipeline, but that is something you can reveal or not.
Yeah, most of the products that we're working on today are with partners, and we're bound by confidentiality. I think the ones that are public, we have. If you go to amyris.com, we have our products listed there that are public. I think we've only mentioned a handful of the 13 today. So you can start to see that the diversity is growing in our products, and we're really excited about that because, you know, we're scientists at heart, and more diverse the more diverse the chemicals are that we produce, the more interesting science we get to do. So I think that would be a good place to start to see the molecules that we have produced.
Yes, because Marcus was asking that, what are the newest molecules for scaling up, and also about your target areas of application. They can raise from, or the application areas. You are speaking about the sweetener. You referred farnesene. You are regardless the application area, you have an amount of molecules planned to be launched in the commercial market soon. And of course, you have confidentiality issues about that. One more to close. Let me see here. It's again about targets. How many advanced biofuel targets? This question is directed to biofuel targets that you have worked in the last year. And how many candidates remain? What scale has project advanced to? What are the applications? It's again more or less the same question. Curiosity about your targets, and in this case related to biofuel.
Yeah, I see the question here in the chat. We have had a long-standing and very good partnership with various branches of the U.S. military who are interested in specialty fuels. So not the standard kind of jet fuel that's used by commercial airliners or the diesel fuel that's used in passenger vehicles and shipping trucks or distribution trucks like semi-trucks. This partnership that we've had with the U.S. military is continuing and it's ongoing. And we have, over the past year, continued to deliver to the U.S. military a variety of different fermentation products they are evaluating for various specialty fuels applications. And at the moment, that's the most promising way for Amyris to pursue biofuels, because those specialty fuels have very kind of stringent and advanced performance criteria and higher price points, which is more commercially attainable in the short term.
Mm-hmm. Okay, it was clear answer. Thank you. Okay, I think we can close the session. I'm very pleased and thankful for, again, for your talk, the audience, great audience that we had today. Great questions also, and great answers. Thank you. Thank you so much, Kirsten and Michael, again. Thanks for this time with us. And for my side, that's all. So, congratulations for your work, and I hope you receive you here one day in Portugal. This is my invitation for you to come to visit, and perhaps giving here a talk face to face. It will be very nice. As you see, you can generate a lot of curiosity and questions. So. Come here when possible to visiting us. Thank you very much. Thank you to all for attending this meeting, and bye to all.
Okay.
Thank you.
Bye. Thank you so much.
Thank you for your time. Bye.
Bye-bye.
Recording stopped.